Hello, everyone, and thank you for joining me for some thoughts on targeted sequencing on large and small viruses. They will be fairly technically because I think there is a lot of detail that people often forget about. Before we start, let me present disclosures. Funding is always important. It's by the National Institute of Health and the state of North Carolina and the Centers for Disease Control. This is another disclosure with respect specifically to Thermo Fisher. Let me begin by telling you about our sequencing experience using the Ion Torrent system and using two viruses, SARS coronavirus 2. We were the first to show the introduction of the G614 allele into the state of North Carolina, and that it came from Europe rather than from Washington State. And then a more novel study published this year on Kaposi sarcoma-associated herpes virus, which is a large tumor virus that we've been working on for a number of years. I picked these two viruses because they're very different, and that will make the point of how do we use a common system to look at very different targets. So SARS coronavirus 2, of course, is an RNA virus. It's relatively small. It has 30,000 base pairs as a genome, and it accumulates to very high titers of roughly 10 to the 8 genome copies per mil in an actively shedding patient. Kaposi sarcoma-associated herpes virus is the cause of Kaposi sarcoma, which is a cancer that's highly prevalent in sub-Saharan Africa and that came to prominence in the US because it was the telltale sign of AIDS in the 1980s. This is a DNA virus. It is much, much larger, on the order of 140,000 base pairs, and it never really replicates. So we're lucky if we get 10 to the 3, 10 to the 4 genome copies per mil in blood. I want to start, however, by going back even further. When back in 2010, we used a PCR tiling approach and Sanger sequencing to sequence West Nile virus. West Nile virus was introduced around that time into the US. And what you can see here are panels A and B. Panel A shows a agarose gel of a number of primers, each covering a 500 base pair part of West Nile virus, because at the time we were concerned that any one primer would fail if the virus mutates, and that's really a problem with RNA viruses. And then we designed these primers so they would work equivalently well at the same melting temperature. And because of that, what you can see in panel B is that you can combine them freely to generate larger amplicons. And that is really the basis for targeted PCR sequencing, called AmpliSeq by Thermo, where you take, again, PCR amplicons that tile the entire genome. And I put up the genome of Kaposi sarcoma virus, which is roughly 140,000 base pairs. And we were able, together with Thermo, to design an AmpliSeq set that in a single tube covers the entire genome in 400 base pair pieces. There were only 5 or 6 gaps which are indicated in the blue. And these are where you expect them to be at the origins of replication, which are repeated GC-rich DNA and then some other repeats in the genome. So what do we need to consider when we are doing these experiments? I will show you that AmpliSeq outperforms random RNA-seq. I will show you that viral load determines the coverage of sequencing, and that the length of each amplicon determines the sensitivity of the approach. I'll show you evidence of a primer bias in amplification, and there's an evidence of a primer bias in sequencing, and that's really where AmpliSeq is different from all other approaches in that it enzymatically digests primers before library build. And I will show you that there are always gaps in any targeted amplification design. The methods are the same. We get the samples in, we inactivate them, we use Roche to make either DNA or RNA. This is automated. We run the AmpliSeq initially on the Ion Torrent S5, now on the Ion Torrent Genexus system. And then we follow up either with PCR cloning and Sanger sequencing or with straight PCR Sanger sequencing. And then we use a number of algorithms to assemble the genome. I'm not going to talk about bioinformatic methods much today. This is the evidence that AmpliSeq can deliver greater coverage and more even coverage rather than random DNA-seq from the Cell Reports paper of 2020. The top left shows RNA-seq, and then the other panels show AmpliSeq in more and more diluted samples. And you can very simply see that there's about a 2-log improvement in coverage, giving the same input. And then you can see that the red lines are much straighter compared to the RNA-seq panel A, attesting to even coverage. I do want to highlight the two ends and draw your attention to the drop in the blue lines at the very 3' and 5' end, and that is, again, one region that any approach never really covers. We were worried about the accuracy of AmpliSeq, so we obtained a sample that is mutant. It's on the left, MT, and it has 428 single nucleotide variants in it compared to the wild type, which at the time had 8. And you can see that over a range of 1 to 10,000 dilutions, we're able to accurately predict pretty much all of the variants, and that is important, obviously. B, viral load determines sequence coverage. And the CDC actually has guidelines that suggest that a CT of less than 28 by any system is required before they will submit a sample to sequencing. The panels here show you the relationship between the CT value and the number of mapped reads on a log 10 scale. If you look on the left panel, these are the CDC primers for SARS-CoV-2. The coloring shows the result of a clear test for positive and negative. And again, if you look at the RNase-P panel, there is no relationship because RNase-P is not related to viral load. If you look at the N1 panel, you can see that there is a linear relationship, very strictly linear if the CT value is less than 30. Between 30 and 40, it's sort of hit or miss, and also a rapid, more than linear drop in log coverage. And on the right, you can see that, again, it's a very nice sigmoidal curve that says percent coverage on the vertical axis versus log 10 mapped reads on the horizontal axis. And as soon as you get around 10 to the 5 mapped reach, you get close to 100% coverage, and 10 to the 5, 10 to the 6, it's that CT of 28. So it really works very well. We did get a lot of samples with a CT greater than 35, and here's an example of 12 of them. Now, obviously, there is background, and the white ones I would classify as random maps, and we don't know if that's library prep or bioinformatics. But the yellow underlaid panels, which again show you coverage at position, clearly are different, and they show that we can use next generation sequencing to detect viruses. It's just much more expensive. But any sample that has at least, 50 to 100 reads, we would declare positive even though we don't get complete coverage. This is again from an older paper where we systematically tried to correlate the amplicon lengths with the CT values at the same temperatures. And you can see that up to about 669, PCR performance is roughly the same. And then if you go into amplicons that are larger than 600 base pairs, you see a linear relationship as the PCR performance drops out under these conditions. I also indicated the mean length of the amplicon either for the SARS-CoV-2 ampli-seq from thermo, the SARS-CoV-1, that should both be SARS-CoV-2, from Arctic, which uses larger one, and then the KSHV ampli-seq that we designed, which are both less than 400. The reason we picked 400 is because that is the maximal read length of the S5 system at the time, and still is. People have looked at that even for longer times, and that is the relationship between PCR sensitivity and amplicon size. If you look at RNA-seq random primed, their fragments are between 50 and 800 nucleotides. If you look at ampli-seq, the average is 200, and Arctic, as I mentioned, is 400. And they're good enough to detect 10,000 copies of target. Now look at the CDC PCR primers. They're roughly 100 nucleotide, and they're certified to calculate down to 1,000 copies per mil. And compare that to PCR amplification for HIV. It took the community 5 years to develop an HIV viral load assay that is sensitive to 400 copies per mil. It took another 5 years to drop that down to 40 copies, and after 15 years, we're able to detect 4 copies of HIV routinely and in a clinical context. And I think where we are with sequencing is really in the first 5 years. We're good if we can detect down to 400 copies, and I hope it takes us less than 10 years to go down to 4 copies per mil. And obviously you can play the same game here with HPV 1618, also a clinical viral load assay, where the most sensitive primers are the smallest. When we looked at failure, most of it happened at the initial PCR step. If we here we're looking at repeated runs on the same target, and you can see that in most cases, targets that don't amplify the first time fail to amplify the second time. The 4 arrows show you sort of the 4 cases. The top left shows both work, the top right shows both fail. The one in the middle is the one case where redoing the initial PCR helped, and then the one on the right, again, redoing the initial PCR helped. Primer bias is a dramatically important piece. We again explored that in diagnostic qPCR. If you remember how a TaqMan assay works, there are 2 primers on the outside and a single detection primers in the middle, and any mutation in that detection primer would influence diagnostic sensitivity. And we measured that directly by comparing the CT values on the right now between an assay run with the same outside primers but a detection primer, and that's on the vertical axis, or run with just cyber green. And you can very nicely see that by relying on the extra probe, you get specificity, but you lose about a third of the positions entirely. That is, mutations in these positions will disrupt the assay entirely. And another third, you lose about a log of sensitivity. And so that always has troubled us. And that is the reason by which each SARS-CoV-2 strain, particularly in the Omicron, we need to redesign the amplification library. This is something I talked about before, in that because ampli-seq depends on primers, the ends are always missing. The 5' end you can only get with race procedures. The 3' end you only get with race procedures because you have to anneal primers at a much lower temperature than other primers simply because the 5' end, if it exists, has a poly A tail. What is an annoying practice lately is that people simply fill in from reference and then don't annotate the sequence. So on the bottom panel, you see the 3' end of SARS-CoV-2, and it looks exactly the same. And in panel A, you see the 5' end, and it looks exactly the same. What we don't know is if that is in fact the sequence of the anchoring primer made to the 2019 Wuhan strain, or whether it's just sequence copied in, or whether it's really 5' raised. And so we can not really support the notion that the utter ends are highly conserved if the data are not annotated. This shows our results where we find that the ends really do matter. We obtained one mutant, which is on the top left, that had a deletion in the 3' end of SARS-CoV-2. You can see the reads piling up. We then, to verify that deletion, took it out of the consensus sequence to generate the 45512 delta target. And now if we align, we have gap spanning reads. And we verify that with Sanger sequencing. And if you look at the predicted M-fold structure in the middle, you can see the deletion is that yellow part. And if it's deleted, much of the structure, in fact, remains exactly the same and retains that functionality. So the fold is not completely abolished, and I think that's why that deletion was viable, even though it's a pretty large deletion. And this is just a reminder that for DNA viruses, you actually need to do Maxim Gilbert sequencing, which most people don't know anymore, to get the 5' end. Coming back to case-HV, and that reinforces the notion that even though we have really good ampli-seq design and really high coverage, there are gaps, and these gaps are functionally related. If you look on the way bottom, you can see the regions in purple where we could not find and place an amplicon. And they map very nicely to repeat regions on the left and to the right, the origin of replication. All of these places have drastically variant GC content. And so amplification will not happen under the same conditions that the other 900 primers amplify that are directed against about 50% GC-rich regions. We had to make gap-spanning primers for Sanger sequencing. You can see that they amplify. You can see they amplify larger fragments. And you can see that not all the amplifications are with the same efficiency. If you compare the bands 3 and 4, 4 is much more efficient than 3. The good news is all of those gap spanning amplicons are less than 1.6 kb, so they can be spanned with the 800 base pair Sanger reads if we sequence from either end. And that's what we did. So if you look at these different technologies and you look at the quality, you can see that Sanger still beats everyone hands down. It's a Q40. You can see that it covers repeats up to 800 base pairs, whereas Dittmer goes to 150 and ion torrent goes to 400, unless you really push it to 800. And then Oxford Nanopore, of course, has really long reads, but you do have other problems, and I'll get into that next. In fact, we compared on case-HV, which is 100,000 base pair virus, what coverage do we get with Oxford Nanopore as opposed to ion torrent? And it turns out that the regions that didn't amplify with ion torrent failed to amplify and give good sequence with Oxford Nanopore. And I think what's different here is that the sequencing polymerase only works on one fixed condition for every region of the genome or the pore for Nanopore. Whereas in Sanger sequencing, we can play around with the polymerase, interested in the 800 base pairs that we want to cover, and we have tricks such as cloning before amplifying and modified nucleotides for GC compression. Lastly, I want to spend 3 minutes on the analysis of virus sequences and the coverage that's needed. if we want to find minor variants, which is what HIV research is about, you need about 2,000-fold minimum coverage. We can get a complete genome by about 20-fold coverage. But more importantly, we can get high confidence SNP maps with just 2x minimum coverage. And that is because 2x minimum coverage across the whole genome still has regions of up to 50-fold coverage for individual SNPs. And if these SNPs are covered, we can use them to generate trees. And here's the example of SARS-CoV-2. There's a bunch of samples that we isolated. They're arranged in a tree. We were able to obtain 48 complete genomes to the specification of GIS8, and we were able to align a tree. But if we looked at a single nucleotide variant calls directly out of the genexus, we were able to actually assemble 67 genomes and samples and order them in the tree. And that's essentially what you're doing with oncology. We are never sequencing the whole genome. You just sequence informative SNPs. So there's a way for this to also work for viruses. And then lastly, just to show you how useful whole virus sequencing is, here we are comparing not just Spike, which is 3,000 base pairs, or SARS-CoV-2, which is 30,000 base pairs. No, we're comparing the entire sequence of KSHV, which is about 137,000 base pairs, of which 90,000 base pairs are unique. And that allows us a degree of resolution that we wouldn't get without whole genome sequencing. So finally, let me acknowledge my lab. We did a lot of that before vaccination was able and available, and that's on the left. And then ever since everyone became vaccinated, we are truly enjoying our time. And with that, I'm happy to answer any questions.